Hello? Oh, there we go. There we go. All right. Good morning. If you're here, present for the day, if you would not mind, please let me know in the chat. Okay, everybody's here. Okay, so we left off yesterday. We were doing a review of mitosis. And we had discussed the idea of, well, the central dogma of biology, protein synthesis, transcription and translation. I believe that I left the mitosis review questions, including this little video here, for this morning. And I said I was going to give you some time in the morning to work on those. I'm trying to think about like a reasonable amount of time to complete these questions. All right, let's go over them again. So you've got it's like 5 questions plus that little video that you'll need to watch for the last one. So why don't we say 30 minutes? That's probably reasonable. I don't normally do this, but we got a touch behind yesterday, and I didn't want to like just dump all this stuff onto you to work on yesterday. So I'm gonna give you half an hour to work on it now. The questions I'm talking about here are the ones on the cell cycle. Some of you might have done them already. I'm just gonna throw it up on my screen here so you know what I'm talking about. It's these questions right here. So I'm going to give you the next half hour to work on these. And then we're going to come back and we're going to talk about meiosis. If you have not already watched it, you should watch the introduction to meiosis video at the bottom here. I mentioned that you should probably do that yesterday, but if you didn't get a chance, this is going to be an important introduction to the process that we're going to go through today. So yes, protein transcription and translation, there's a lot of complexity to that idea, but believe it or not, it is not the most complex process we're going to talk about in this unit. The most complex process is definitely meiosis, and this is the one thing that students in this course typically struggle with a little bit. So we're going to spend quite a bit of time on it, or at least as much time as we can with this type of scheduling. We'll do a review of it tomorrow as well, because this tends to be the process that is the most confusing for grade 11 students. And I have to be perfectly honest with you, like 9 years ago, when I started teaching, I taught grade 11 biology that first year that I taught, and I hadn't looked at meiosis for a long time, probably since I was in high school. It didn't really come up in university. I didn't take any cell bio or stuff where meiosis was really integral. And I had to relearn it. And yeah, I found it a little bit complicated. I'm not going to lie. It took me a minute. So, and I didn't have these cool video resources and stuff like that to help. They actually make it quite a bit easier to understand. But yeah, this is definitely the most complicated process that we're going to examine in this course. So I highly recommend that you take a quick look at this video before we go through it together, and you can watch it afterwards again because it might be helpful. But we're going to go through it step by step. It's similar to mitosis in that it is a step-by-step process for how a cell goes through a division, and meiosis is actually 2 divisions, but instead of making exact copies, which is what happens in mitosis, meiosis is all about making sperm and eggs. So it's a slightly modified version of mitosis for making sperm and eggs. And there's quite a bit more going on there. So anyway, I'm going to let you guys work on that. Half an hour would be basically 9 o'clock. So I'm going to come back at 9. Okay, guys, let me know if you need anything. And oh, actually, before we go away, does anybody have any questions from the content from yesterday? I should throw that out there. So these are a number of people came to visit me in the Google Meet, which is great. Also, some people just drop by the Google Meet to talk about cool biology stuff. Thanks for doing that. I appreciate it. That's fun. Will you be looking at our presentations as PowerPoints or Google Slides? So yeah, how Brightspace does it, which is a bit of a bummer, is that it converts your Google Slides into a PowerPoint, and then from PowerPoint, it converts it into a PDF. So what I'm actually looking at is a PDF of a PowerPoint of your presentation. So in that conversion, there are minor formatting weird things that happen. Stuff gets shifted around, fonts get changed. For them, I ignore that stuff. I mean, what can I do? That's how the software works. It's not great. So like minor things, like you have some words moved around or it makes it a little bit hard to read. I assume that in your original, it doesn't look like that. My assumption is that that is part of the conversion process, that it's like putting text over text or moving text slightly off or askew. I'm assuming it's part of that conversion. So it does look a little bit different. Don't worry about it. But basically don't worry about it, because I just ignore that minor formatting stuff. I'm looking for like the key components of the presentation. Now, sometimes the formatting changes actually make it difficult to use. Like, for example, it might make your dichotomous key like difficult to understand what you're supposed to do. Like it'll move the lines around if you did a visual dichotomous key or something like that. You can always check, because if you convert yours into a PDF and then look at it, you'll see what it looks like. Or you can even, well, after you hand it in, you can look at the assignment handed in to see what it looks like. Yeah, so you can also do that. You can just convert your presentation directly into a PDF and then hand in the PDF. If you do that, it's going to look a lot more like your original. So yeah, you're welcome to do that. That's actually probably helpful for me. And it doesn't change how I view your presentation in any way. So it doesn't, like the format doesn't have to be Google Slides. It needs to be in the form of a Google Slides presentation, but it doesn't need to be that file format. So anyway, sorry, hopefully that answered your question. I basically ignore all the small little stuff though, because it's just not relevant. So any questions about mitosis, protein synthesis, transcription, translation, anything? Let me know. I'm happy to go over anything. If you didn't get a chance to write your quiz yesterday, please find some time today to write your quiz. At some point today, it definitely needs to get done. Some people emailed me about extensions on that stuff. If we agreed on a different timeline, then yes, obviously, whatever we agreed upon, just honor the agreed upon timeline. Absolutely, Alex. Resubmit it. I'll be able to see that you have an original submission that is before the deadline, and it won't matter. It won't matter. Absolutely submit it. Okay, I'm going to assume that there are no other questions, unless some people are typing. It doesn't tell me that. Oh, just somebody showing up. Okay, cool. So if you guys need anything, don't be afraid to let me know, and I will see you at 9. All right. Let's get back to it. So, at this point, hopefully you have reviewed mitosis and have looked at the meiosis summary. What we're going to do now is look at that process of meiosis in more detail. Does anybody have any questions from these questions, the mitosis review questions? If you do, throw it out there. You're going to need the next package as well, I believe. Yeah, it's right here. Right above this meiosis video, there's a little post that says part 2 of the course notes. So that's the those are the course notes that we're going to be using for this next part. So you're going to want to grab a copy of those and open it up in Cami, or if you have the package printed off. Oh, excuse me. That's just the next part in the complete package that I sent out at the beginning of the course. So I'll give people a second to pull it up. Oh, sorry, I didn't have my chat scrolled all the way down, and I missed this. Yes, you are correct, Andrea. That's what they do. So the spindle fibers are the structures that actually physically pull the sister chromatids apart in mitosis, and they are going to be involved quite a bit in meiosis as well. They are able to reposition molecules within the cell. And this is one function for spindle fibers, or microtubules, which is what spindle fibers are made out of. It's a specific type of molecule. And microtubules are actually used for moving other structures around the cell as well. But pretty much in high school, the only function for them that you will care about is in mitosis. Thanks for answering that question, by the way. I got to keep an eye on that. I didn't have my chat scroll down all the way to the bottom because I went up to double check my attendance. Okay, so in the meiotic process, sort of the big idea, the main thing that your main takeaway here is that a lot of the diversity in sexually reproducing organisms comes from this process that we're about to look at. And by diversity, what I mean is if two members of a population, use your parents as an example, if they have multiple offspring, so more than one, no two, unless you are in the very unlikely situation of having identical twins. But other than that, no two offspring will be the same. They will have a different mix of the parents' genomes, mother and father. And that is a result of meiosis. So meiosis ensures that every time you make an offspring, you make a different one, in terms of a different mix of your genes going to your offspring and a different mix of your partner's genes. And what that ends up creating is diversity within one generation. All of the members are basically a mix-up of the previous members. And that is useful. I mean, we went over this in the diversity unit about why genetic diversity is useful, but essentially it means that the species is more resistant to external environmental stresses. You're going to get members of the population that are able to deal with higher temperatures, able to deal with lower temperatures, able to deal with a specific, I mean, honestly, you name the environmental condition and there's a range of values that are acceptable to a population. Some people can survive with less water. Some people are better at storing fat. Some people are better at burning fat. So, I mean, like, you pick an environmental condition, and there's going to be variability in the population in terms of how you respond to that environmental factor. And so the more diversity you have in a population genetically, the more likely you are to survive. That's sort of the main thrust of the diversity unit. And meiosis plays a role there. It is the reason that variability exists. The video of the second half of meiosis was pretty similar to mitosis. Absolutely it is, Owen. The second meiosis 2 is basically mitosis. That's exactly what it is. So I mean, obviously there are some minor differences, but overall, biochemically, it looks exactly the same. If you were to watch Meiosis 2 under the microscope and watch Mitosis under the microscope, they would be indistinguishable. You actually wouldn't be able to tell them apart from each other. So yes, absolutely. Anyway, so that's the main thrust of this. So I'm going to mention a number of times throughout this process as we look at meiosis, oh, hey, here's an area where you increase genetic diversity. And we've already talked about why that's important. And so take note of that. Take note of that because, again, that's the purpose of this. Why don't we just make clones? We could do that as humans. I mean, there are species, animal species, that clonally reproduce. They have an egg, and it just has all of the DNA of the parent, and then the offspring is the same as the parent. You can do that in nature. Organisms do that. It's just not a good reproductive strategy because it does not encourage diversity. And when you remove genetic diversity, you decrease your ability to survive in environmental stress, which is why it's not favored. So if you look at the natural world, most species sexually reproduce because in most situations it's favorable, even though it's way more energy intensive and complicated. And despite all of those things, you have to find a partner. You need to go to the trouble of making these complex gametes. I mean, all of this stuff takes energy and is complicated and can fail. We're going to talk about some ways where the process can fail later. But the overall benefit is that it just increases diversity. So there's I added some terminology in here. I crossed out one word here, which was non-sister chromatids. Again, these are sort of riffs on some notes that we share in the department. I hate that term, non-sister chromatid, because it doesn't, well, it's not real. It's not an actual biological term. We talk about this idea of sister chromatids in mitosis, and that exists also in meiosis. There are sister chromatids that are basically identical copies. But at some point during mitosis, meiosis rather, sorry, I better be careful about keeping those two straight. At some point in meiosis, you modify the chromatids so that the two sisters are not identical to each other. But they're not non-sisters. They're still sisters, but they're just not identical any longer. So I changed that in my note to non-identical sister chromatids. That's a much better way of thinking about it, in my opinion. I'll show you later where that comes in. Okay, so again, what are we doing here? You need to pass on your genes to your offspring. That is the purpose of mating. And from an evolutionary perspective, that is the whole reason you do it. It's why you're motivated to do it. It's why psychologically humans are programmed to enjoy the mating process. And not just humans, by the way. All organisms are programmed to be drawn towards the mating process. And that's for survival of the species. Basically, you need to compel organisms to do this process because while it allows them to pass on their genes, it is a pain. I mean, it is a. I mean, okay, we don't need to get into details here, but the birthing process, there are a lot of things associated with child rearing that are difficult. They're not necessarily the things that people would readily do. And so, yes, nature sort of drives organisms towards this process, even though it's complicated and difficult. But it's necessary to pass on our genes. So, anyway, if you ever take an evolutionary psychology course or something like that, there are some fascinating stuff. There's some fascinating stuff being discussed there in terms of where certain human behaviors come from and how they're genetically and evolutionarily advantageous. And if you have a fascinating course you could take in university if you're interested, but I won't get too deeply into that. We're making gametes. That's what we're doing here. Gametes are sex cells. And I mentioned previously that those are sperm and egg. Okay, so you got to make those. And in the process of that, we have to take a diploid cell, which is what we're going to start with, a 2N cell, which in humans is 46 chromosomes, and we have to cut the number in half. You only get to give your offspring half of your genetic information, and your partner is going to give half of theirs. And that's true for all sexually reproducing species. They're going to give half to their offspring. So we need to take an adult cell. We're going to start with an adult cell. Adult cells in your body are called somatic cells. Almost all of the cells in your body are somatic except for your gametes, which means that they have the full genetic complement of information. They're a regular adult cell that just does mitosis all the time. Mitosis is what's happening in all your cells. They're doing reproduction to make exact copies of themselves. Okay, so those are somatic cells. Sometimes they're called adult cells, but you'll probably never see the word adult cell outside of here. They're pretty much exclusively called somatic in biology. Okay, and our diploid number is 46. I already mentioned that. So if you were to take a somatic cell, one of your skin cells, for example, and a somatic cell from a partner, their skin cell, whatever, I mean, you could pick anything, and merge them together and be like, well, there you go. Like, let's just take two adult cells and put them together. Why don't we just do that? Well, you're going to end up with double the amount of genetic information that you normally would want inside of a cell. And so going back to what we talked about yesterday, which was protein synthesis, imagine that the nucleus now has double the amount of genetic information. So every time that you're transcribing a particular gene, let's say the one for insulin or the one for testosterone or the one for whatever, pick an enzyme, you are always doing it twice as much. Okay, so you're doubling the rate of every gene being expressed. You can quickly see how that would be untenable. Not only do the materials not exist to double the transcription of every gene, but it's going to disrupt every biochemical process inside the cell. Essentially, cells cannot survive if you ask them to double their output of everything. It's not possible for them to be alive like that. So that situation where a cell is supposed to be diploid, but ends up with more than diploid, is called polyploidy. Poly just means many, and ploidy is just the amount of genetic material in the cell. So diploidy, or diploid, just means that you have two of each chromosome. Okay, a maternal and a paternal. And diploid, or diploidy, is the standard for a somatic cell in a sexually reproducing species. Now, of course, there's always exceptions to this. There are some species that operate on greater than diploid. So they have more than two copies of each chromosome, and they're fine with that. So this is confusing because later on you might look up chromosome numbers and be like, that doesn't make sense. That number seems inappropriate for this species. Like, how does that work? It's because they're not actually diploid. So a good example would be a number of plants actually operate as tetraploid, which means that they have 4 copies of each chromosome, 2 from the mother and 2 from the father. But that's an exception, okay? That's by far not the most common in nature. Diploidy, or 2 copies, one from the maternal and one from paternal for each chromosome, is what's typical in nature, okay? So I'm going to describe diploid all throughout this here as being the norm. Diploid, 2 of each chromosome, one from mom, one from dad. That's the normal number, okay? Or the 2n number, diploid. And I'm going to also describe polyploidy as being incorrect, okay? Not desired in terms of an adult organism. So that is typical in nature, but I just wanted people to be aware that there are some exceptions to that rule. And so I don't want them to be confused later. You might come across this, especially when we're doing our project at the end of this, and be like, I don't understand what's happening in this situation. That's what's happening, is that it's more than diploid is normal for that organism. But again, it's not typical. So so basically what happens if you get polyploidy, that is, you have 3 or more sets of chromosomes, okay? So you could also have 3, or 4 or 5 or 6. Triploid would be 3. Usually it results in some type of developmental abnormality or death. Keeping in mind that when you were talking about polyploidy, you're talking about there being many of the chromosome, okay? And it could be 3 or 4 or 5 or 6. That polyploidy just means many. There's another word for this. This is going to get confusing. This comes up a little bit later. That just means the wrong number, okay? The wrong number, or the incorrect ploidy, is called aneuploidy. Oh, did I spell that correctly? No, and up loudly. Autocorrect. Aneuploidy. There we go. Aneuploidy. So polyploidy means many, but aneuploidy, the word aneuploidy specifically means wrong. So whenever you see aneuploid, that means incorrect number. Whatever that number is, it's not the right number. It's not what's supposed to be there. Okay. Whereas poly doesn't necessarily imply it's incorrect. It just means that there's many. Okay. So these are like little minor language things that you're going to run into. Okay. So what do we want to do when we reproduce? Well, we want to produce a gamete. The partner wants to produce a gamete. And so then we're going to fuse those gametes together, fuse the specialized sex cells or gametes together to make a zygote. A zygote is a gamete from one organism, one member of a population, and another gamete from another member. Okay, so there are two versions of this. This terminology here is specific to humans, but a similar style of process exists in other organisms. Actually, I shouldn't say humans. This is specific to animals. Not humans necessarily, but animals. If you go into the other kingdoms, often there are similar processes. They have different names, but they essentially are exactly the same as this. Okay, so but keep in mind that what we're talking about here is mostly from a human perspective. So spermatogenesis is meiosis. It's the meiotic process for males. And I mentioned that you start off with a 2N cell, a somatic cell. And that somatic cell, whoops-a-daisy, just deleted my E. The somatic cell is called a spermatocyte. That's what you start with in the testicles. There is, in the seminiferous tubes, that's where you find spermatocytes. So that's the starter cell, and it's a regular somatic adult cell that has the full complement of genetic information. And you're going to take that spermatocyte and you're going to build sperm. That's what you end up with at the end, or spermatozoa is the full name for them. Okay, so it goes from spermatocyte to spermatozoa. There's some math involved here, in that you start off with one spermatocyte and you end up with 4 sperm at the end. Okay, and we'll talk about how you get 4 from 1, but that's how the math breaks down. So you have one 2N cell, one diploid cell, and you end up with 4 haploid cells with half of the amount of genetic information. For females, the process is called Oogenesis or Oogenesis. Which one of those is correct? You'd have to ask whichever prof is teaching you the course on that particular day, because I've heard this pronounced multiple ways. I asked my wife, and my wife said Oogenesis, and then I asked another friend of mine who's a biology prof, and he said Oogenesis. So take your pick, however you want to pronounce that, but oo is very common. So it's either Oogenesis or Oogenesis. That's the meiotic process for females. Okay, so they're specialized used to make your specialized sex cells for women. The cell that you start with at the beginning of the process is called an oocyte or oocyte, again, depending on who you ask. So you start with an oocyte, you go through meiosis, and you produce an ootid. An ootid is also occasionally called an egg. Okay? That's the same thing. So, I mean, I'm sure you've heard of females producing eggs. That's what it is. So the science name for that is an ootid. So an oocyte or an oocyte makes an ootid. One oocyte produces one ootid. So you might be looking at that and being like, well, that's weird. It's the same process in men, and one spermatocyte produces 4 sperm, but in females, one oocyte produces one ootid. Yes, so there is a minor difference here between spermatogenesis and oogenesis. So we're going to talk about that difference, obviously, and why the math is a little bit different. We'll get to that later. Okay, and where does that happen? This process is taking place in the ovaries for females. And that's true for virtually every single animal has some form of ovary. Although if you go way back to the oldest iterations of animals and you look at the like jellyfish and sea sponges, they have sort of a variation of that. Mostly because they don't have real tissues the way that we do, but they have something similar. Okay, so again, each of the cells that you're going to make, sperm and egg, or ootid and spermatozoa, are going to be haploid. And remember that I keep harping on this, I realize, but that's called 1N. Okay, that's how you annotate, or N, how you annotate the number of chromosomes. Okay, half of the full complement. The reason I harp on it so much is because that's new terminology for this course, and people tend to get confused with haploid and diploid, and it's kind of important. So at the end of the whole process, when male and female come together in the process of fertilization, you're going to end up with one sperm, which is going to have 23 chromosomes in it. That's the haploid number. And one egg, or ootid, that has 23 chromosomes in it, or 1N. And you're going to put them together. They're going to fuse, and they're going to form a diploid, or 2N, zygote. A zygote is a fertilized egg. These are great terms to add to your vocabulary list. Although I think you guys learned this basics of this in health class? Question mark? I'm not sure. I don't remember. I don't know what you guys learned in health class. I learned this in health class when I was in high school, but I don't know how the curriculum has changed since then. Okay. So again, the big idea, the main overarching idea here is that haploid gametes are made to create genetic variation. That's why you make them haploid. And increased variation means better survival for the species. Diversity equals survival is sort of like the theme of the course, kind of. It's in almost every unit. So, all right. So I'm going to go through this process now step by step, the same way we went through the mitotic process. It is virtually identical in spermatogenesis and oogenesis, so the male and female versions, with a very small difference, and I'll talk about the differences when they pop up, but keep in mind that I'm just talking about the general process here that exists in males and females. When you do your project, you are going to choose. So you're going to show me this process. You're going to explain it to me, or to another student via video, okay? And you are going to choose the male or female process, and you're going to specialize your explanation just to this is the male process or this is the female process, and you're going to pick a specific species to show me this process in. So you might say, okay, well, I'm going to pick the yellow fever mosquito, okay? Aedes aegypti, I think it's called. And the reason why you would choose that is you have to pick an organism that has a 2N number, a full number of chromosomes between 6 and 8. And that just happens to be a species that fits in there. So maybe you pick that. That means you're going to show me the right amount of chromosomes for that particular species. And you're going to show me the exact process for females or males or whatever. So just keep that in mind in the back of your head. You're going to be showing me this process later. So Meiosis, if you watched the little video, you would have seen that it's broken up into two halves. Okay? And somebody already mentioned, who was that in the chat? Owen already mentioned that meiosis 2, the second half of this process, basically is mitosis. It looks exactly like mitosis. So, whew, if you understand mitosis, the second half of this is a breeze. It's the first half of the process that always gets people. So, meiosis 1, the first half, is called reduction division. Okay, and the reason that meiosis 1 is called that is because you are reducing the amount of genetic information in the cell by separating it into two. That is the objective of the first meiosis. Okay? Keep in mind that there's two that make up all of meiosis. There's meiosis 1 and meiosis 2. Okay, and the phases have the same names as they do in mitosis, and they are similar to the phases of mitosis, but there is always a number after. So it will be called interphase 1, prophase 1, metaphase 1, anaphase 1, cytokinesis 1. Interphase 2, prophase 2, metaphase 2, anaphase 2, cytokinesis 2. There's always a number afterwards. So if you see just the word interphase, interphase is mitosis. Interphase 1 is meiosis. Okay, so there's always that number there. And when you're doing your version of this and showing me, don't forget to label them as interphase 1 or interphase 2 because they're different from each other, and they're also different from mitosis. So there is no just plain metaphase in meiosis. There's metaphase 1 and metaphase 2. Okay, so just to be clear, here we are. We've got our spermatocyte or oocyte, our starter cell. And what happens during interphase? Well, it's very similar to what happens during interphase in mitosis, which is that you grow and you build the specific structures that are needed for cell division, okay? And most importantly for us, you double the DNA. So you make an exact copy of every chromosome. Okay, that is the same as mitosis. You're making an exact copy of every chromosome. Okay? So you copy, and you're making copies of the centrioles for growing spindle fibers. So what's happening here is that we started with a 2N cell. Okay? And now, after that, just like in mitosis, we have 2 times 2N. But really important to note here, it is not 4N. Okay? Not 4N. 4N would imply that you have doubled the amount of information present. And if you make a copy of a book, you don't double the amount of information you have. You only double the amount of books that you have. And so the exact same thing has happened here. We have the same number of books, we have the same number of information, the actual information in the DNA, but we have double the amount of it. Okay? So you cannot write this as 4N, and you also can't say that there are 92 chromosomes, because there are not 92 chromosomes here. There's 46, because a chromosome implies unique genetic information. Okay? So there's 92 pieces of DNA, but there's still only 46 chromosomes. All right? So you would still call this diploid. All right? This is really confusing for students because you've doubled the amount of things in it, but it's still diploid at the end. It's just 92 pieces of DNA. Oh, I'm going to spell pieces wrong. I'm one of those people that always mixes up the E-I-I-E thing. I don't know what that is. I don't know why I always do it. I feel like I do it every single time I write these words. I want to put I-E. Is it I-E? It's going to look weird. Is that correct? God, I feel like I do that every time I write these words. I'm 36 years old and I still don't get this right. What's wrong with me? Is it I-E? Pieces? Pieces? This is one of these things where I really should have just figured that out. P-E-I? No. Wait, what? P-I-E. Yeah, it is P-I-E. Okay, I did it right there. Okay. Oy, oy, oy. That's sad. Okay. Anyway, 92 pieces of DNA. But it's still diploid. All right. So all we've done is duplicated the DNA. Same as interphase in mitosis. Next. Thank you. Appreciate it, Macaulay. Okay, next. Just like mitosis, we're going to do prophase. So the order of the steps and the names of the steps are very similar, okay? So if you'll remember in prophase, what happens in mitosis, a lot of it is the same. So first of all, you bust that nucleus apart, okay? That happens in prophase of mitosis as well. You also take that long loopy DNA that's all laying out in the cell called chromatin, and you wind it up into DNA sausages, okay? So that exact same process happens. That's called condensation. And you condense the DNA into chromosomes. So that is exactly the same as mitosis. And the centrioles, you can see them right here, are going to start creating spindle fibers, and they're going to start moving to the opposite poles of the cell. Again, exactly the same as prophase of mitosis. Okay, so that's the same. Where things start to differ is that in this, okay, so here we go. Here we have our sisters. Remember that sisters are identical and they're attached to each other. So the two blue ones here are sisters and the two pink ones here are sisters. Okay? But those two are homologs to each other. Remember that every chromosome has a second, I don't want to call them copies. They're not copies. A second one that has the same information on it, the same genes, but they are different because they're from the mom of that organism, okay, or the dad. So remember, those are called homologs. You have one chromosome 1 from mom and one chromosome 1 from dad, and one chromosome 2 from mom and one chromosome 2 from dad. So this would be like your chromosome 1 from dad and your chromosome 1 from mom, and then you've got another one here where you've got chromosome 2 from dad and chromosome 2 from mom. And those two are not identical to each other. In meiosis, okay, now in prophase 1, the homologs are actually going to get together. Okay, they find each other in the cell and they give each other a hug. Okay, they hug each other. So that process is called synapsis. Synapsis is when homologous chromosomes grab each other and hug it out. Okay? So they grab each other and hug it out, and that structure that they make when there are 4 chromatids basically stuck together, 2 paternal and 2 maternal, is called a tetrad. Okay? And really, tetra means 4. There's 4 chromatids. They're stuck together. Okay, so that structure, that when they're all together here, is called a tetrad. Why do they do that? Well, when they get into a tetrad, they do a process called crossing over, which means genes from one homolog are going to switch with genes from another homolog. So before you draw this, I'm gonna demonstrate this process happening. Okay, so if you can imagine, I've got chromosome 1 from dad, okay, and its identical copy. Chromosome 1 from mom, and its identical copy. Remember, identical copies are called sisters. Okay, so the sisters are attached to each other here. And then there's a centriole in the middle, or somewhere in the middle-ish, that's holding the two sisters together. And we can also look at the actual genes on here, okay? And they're labeled off to the side. We actually use letters to indicate which version of the gene is there. And there are multiple versions. For example, you have a gene for eye color, okay? And let's say there's two versions of it. There's more than two in real life, but let's say that there's two versions of it. A version for brown and a version for blue, okay? So we would use letter combinations to indicate that. So we might say, okay, well, the one you have from your dad is going to be brown. Brown is going to be capital B. And the version that you have from your mom, I don't know why I did this opposite to what's on the other side there. It's kind of annoying. But the one you have from your mom is going to be for blue eyes. And so we're going to represent blue eyes with small b. Now you'll notice that within the same, within the sisters, okay, remember that the two pink ones are sisters and the two blue ones are sisters here. The versions of the gene that are present there are going to be the same. They're always the same because those are identical copies of each other. So one blue one and the other blue one are identical. They're sisters. So whatever I draw on the one blue one, I got to draw on the other blue one because they're exactly the same. And the same thing for the pink one, okay? And so let's say that there's another trait on here, another gene. In real life, there's thousands of these. But let's say that there's another gene for, and it's near the bottom here, and it's the gene for, I mean, there's so many genetic traits I could choose here. We're going to talk about some of these possibly later today, or maybe tomorrow. Attached earlobes, okay? You have a gene that is either going to say that your earlobe down here is either attached to your head or it's a free earlobe, all right? So I'm going to say big A is for attached earlobes, and little a means free earlobes, okay? Whatever, doesn't really matter which trait you pick. So the version of the gene is different on these two ones here, okay? So this version over here and this version over here are different. That's why they're shown with different letters. Not different letters, different like capital and small letter, okay? Different case of the letter. Those versions are called alleles, okay? Spelled like this. This is going to come up later again. We'll write this out again, but I'm going to use the word allele here, so I'm going to just make sure that I mention it. So there are different alleles here. So these are going to give each other a hug. Okay, so we got the paternal and the maternal, and they're going to get together. They're going to hug it out. Oh, didn't like that. Okay, they're going to come in, give each other a hug. Normally I would actually put these right in and make them touch each other. Except why? Why did it just do that? There we go. Normally I'd put them right in, but I got to move some stuff around here. So they're going to hug each other. And the reason that they hug is because they are actually going to exchange some DNA, some genes, between the two of them. Okay? And keep in mind that they are exactly the same in terms of where their genes are located because they're homologs. So all humans are going to have homologs here on chromosome 1. This spot on chromosome 1, which is called a loci, or a locus. Locus is for singular. So this locus, this place on the chromosome codes for this. It's the exact same place on the homolog. Okay? It's the same locus. And again, that's going to become, within the same species, those places are all the same. So we're going to exchange a piece. That guy on there. That guy on there. Okay? And so now when you look at them, the sisters, remember the sisters are like two blue ones or the two pink ones, they're not identical to each other anymore because you've done a little switcheroo. You've switched some of the DNA between the two of them. And so if you look at the actual chromatids themselves, so the first stick of DNA, the second stick, the third stick, and the fourth stick, you'll notice that they are all different from one another. No two sticks of DNA or chromatids are the same any longer. So what did that do? It created diversity. Hooray, diversity means survival. So the more that you do this, the better the survival of your species. Okay? So that process of switching DNA, actually exchanging DNA between homologs, is called crossing over. Probably, arguably, the most important part of meiosis. Okay, so that's why I quintuple highlighted this here. So within tetrads, you're going to get that switching of DNA so that every chromatid is different from every other chromatid. So that process is called crossing over. And why is it called that? Because DNA crosses over between the homologs. It's probably not that confusing why it's called that. But so when the DNA comes in and touches at those various points, that's called synapsis. So much terminology, guys. Synapsis is where they come in and touch, and then crossing over is when they switch. Okay, so synapsis, they form a synapse, touch each other, and then they switch. That's called crossing over. So synapsis and crossing over. And it is a physical exchange of genetic material between homologs. You wouldn't exchange between sisters because sisters are identical to each other. So it's only happening between homologs. When you take a piece of DNA and you modify it by switching up the order of stuff or switching things from one chromosome to the other, in genetics, we call that process genetic recombination. So this is a form of natural genetic recombination. We also do artificial genetic recombination in the lab. We can, for example, take a piece of a gene from a tomato plant and we can put it in a corn plant, or take a gene out of a fish and put it in a tomato plant. And we actually do that stuff all the time. It's really cool because we can give traits from one organism to another organism in order to make them last longer on the shelf, grow faster, produce more sugar, taste sweeter, whatever. Okay, so we can also do that in the lab. But that process of changing DNA, switching one piece of DNA for another piece, is called genetic recombination. So this is just a form of genetic recombination. We also have artificial genetic recombination, but this happens every time you make a sperm or an egg cell. You recombine DNA. When humans do it, it's also something that increases genetic variation, but just artificially. Great question, Owen. So officially here, the two blue ones on the left are still called sisters. That is their actual official name at this point still. Right till the end of the process, they're called sisters. But that is so confusing because the thing that defines them as sisters, that they're identical, is no longer true. And this is exactly where the term non-identical sisters comes from, which I added to the beginning here. I call these non-identical sisters because sisters means identical. But you can't call them non-sisters because they're still sisters to each other. They were made identical. So this is a point of contention between me and other people in the department. But I call them non-identical sisters at this point. They are still technically called sisters, though, which is confusing. I'm not sure if that answered your question or made you more confused, but they are technically called sisters still. But I'm just going to refer to them here as the non-identical sisters. They were identical sisters. Just to keep them straight from homologs. Anyway, so they're still locked up into that tetrad, into that hug. Okay, so the hug continues. They've done the switch, but they're still hugging each other. The homologs are still hugging. Okay, before we move on now to the next step, to metaphase 1, are there any questions about prophase 1? This is the hardest step. This step, the most stuff happens, and it's the most confusing out of the entire process. So pretty much if you get what's going on at this point, you're probably good. But if you don't for some reason, please let me know. Any questions about prophase 1 and what's happening here with crossing over? For those of you that are thinking, okay, I think I get this, but this is complicated and I'm not 100% there yet, that's fine. That's fine. Tomorrow, what I'm going to get you to do at the beginning of class is I'm going to get you to grab some colored pieces of paper, yarn, zip ties, whatever you got, basically. Something to represent chromosomes where we can switch pieces between them. Pipe cleaners works, although not everybody has pipe cleaners at home, but whatever you have, colored pieces of paper work fine because you just need that and some tape. And that's what I'm going to use. So I'm going to go through this process, and you are going to go through it as well on your desk, bed, wherever you're working. I don't know where you are at home, but and we're going to try and go through the entire process together with some physical, manipulable things in front of you to help sort of get this process straight. And that will, I think, it usually helps. And usually after that, I think students usually have a lot fewer questions because it makes more sense. You've done it with your own hands in front of you, and it makes more sense. But don't worry if you're just sort of somewhere on the path right now where you're like, I think I get this, but like, let's see where this is going. Okay. So that's prophase 1. Next comes metaphase 1. Again, just like mitosis, there's a metaphase after. And also just like metaphase, the main thing that's happening here is that you're going to be repositioning your chromosomes in the center of the cell, like on a line. Okay, so just like in that process of mitosis, there is a metaphase plate involved here. Okay, it's right down the center line of the cell. If this was mitosis, you would be putting those sisters, you would, whoopsie daisy, I didn't mean to grab that metaphase plate, but you would be putting the sisters right on the center of the plate where one sister was on one side and one sister was on the other. But that doesn't work because the sisters are still attached to their homologs. They're still in tetrads. So what happens in metaphase 1 is that you're going to take, I didn't mean to grab that metaphase plate, get back there. You're going to take the entire tetrad and you're going to place it on the center so that one non-identical sister is on one side and the other sister is on, oh my goodness, I'm using the completely wrong terminology here. So that one homolog is on one side and the other homolog is on the other. But the sisters, the non-identical sisters, are going to stay together. Okay? So you'll notice that the two pink ones here are together. They are going to stay both on this side. Okay? And for this one, these are going to both stay on this side. They're still locked into their tetrads, but you got basically almost the whole thing on one side and almost the whole other thing on the other side. Okay? So this basically looks like metaphase of anything, except now you've got homologs on either side. One important thing happens during metaphase 1, and that is a process called independent assortment. And what independent assortment means is that how we place these homologs. Am I going to place the sisters on this side, the maternal here, or am I going to place it like this? So is it going to go maternal on the left and paternal on the right, or maternal on the right and paternal on the left? Or again, let's look at another pair. Am I going to put these over here or these over here? Which way is the maternal going to go and which way is the paternal going to go? That is completely random. And it's actually very important that this is random, because what that means is that you're going to get a different mix of things going left and things going right every single time you do this. So one time when you make your egg, chromosome 2 is going to go maternal on the left and paternal on the... Did I just switch that up? Yeah, sorry, maternal on the right and paternal on the left. And maybe the next time you make gametes, you're going to switch it up. Maternal will go left and paternal will go right for chromosome 1. And maybe sometimes it goes this way for chromosome 2 or whatever's up here, and maybe sometimes it goes this way. Which side the maternal and the paternal sisters land on is completely random. So that randomization of which way they go is called independent assortment. And this is the second thing that happens during the process that adds genetic diversity. Because you're basically randomly mixing up maternal and paternal to the left and to the right every single time you do it. Maybe one time you get all maternal on the left and all paternal on the right, and maybe one time you get maternal, paternal, paternal, maternal, maternal, paternal, and maybe one time you get something completely different. Every single time you do this, it's completely random, which means that the mix of genes that you get is completely random every single time. So normally, I mean, like, who cares that you're randomly mixing these? Well, that increases genetic diversity when you mix them. So who cares? Evolution cares. Because it increases genetic diversity in your offspring. It means your offspring are more different every time you make them, which means that your species survives more often. Diversity equals survival. Stretch here. How am I doing for time? Oh, I think we're going to get through 3/4 of this in the first half, which is pretty good. Oh. All right. So I just have this summarized here. Oh, why do I have that written there? Nuclear membrane is broken. No. That happens during prophase. Shoo. How have I not noticed that before? Okay, you break the nuclear membrane in prophase. That's annoying. That shouldn't be there. Though I'm gonna have to go back and fix that. I'm actually really miffed that I missed that. That means that's been in my note for a long time. That's really annoying. I must have been focused on the independent assortment so hard that I never noticed that. But the nuclear membrane broken down goes in prophase. Any questions about this so far, about what's happening? We're lining stuff up. We're lining the pairs up on the plate. Now, I mentioned here at the bottom here that the sisters do not line up on the plate. Sisters do not go on the plate like they do in mitosis. Here we have homologs going on opposite sides of the plate. Okay, so you could say that the tetrad is sitting right on the plate, where one pair of sisters is on the left and the other pair of sisters is on the right. So it's a little bit different from mitosis. Okay, so that's metaphase 1. Now, the next phase is anaphase. This is going to look a lot like mitosis in that the purpose of anaphase, or banana phase, is to pull things apart and put them on opposite ends. But instead of breaking the sisters apart like you do in mitosis, here the sisters are going to stay together. So we're leaving the sisters together, but the homologs are going to break apart from each other. So they are no longer going to be with their homologous pairs. Homologs are going to get pulled apart. Again, these are the big things that students always confuse. So we do not split apart the sisters here, like in mitosis. That's the difference between this and mitosis. Sisters are going to stay together. And it's going to be a mix of paternal and maternal on both sides. That one's fairly straightforward. Okay, now we got telophase. Again, this is going to look pretty much like mitosis in that in telophase, you put a new nucleus around the genetic information, and then it's going to decondense and go back into its chromatin form again. So under the microscope, basically the DNA becomes invisible because it goes back into chromatin, and you can't see those structures under the microscope. So it unwinds and goes back into its chromatin form. You can also see the cleavage furrow right there because cytokinesis actually starts before telophase. So it's kind of happening in the background during telophase. The reason I put it at the end is because it finishes after telophase, and so that's where I'm putting it in the process here, but it overlaps. So you can see the cleavage furrow, again, just like you would in mitosis. And then here we are at the end. Cytokinesis is happening, and keep in mind that it's called cytokinesis 1. I forgot to include that in my note, geez. But yes, they all have numbers after them. Cytokinesis 1, you're going to split apart your two cells. Now, at this point, the chromosomes in each of these cells do not have homologous pairs. So they don't have a maternal version and a paternal version. So you would say that each of these are now haploid. They do technically have 46 pieces of DNA, but each piece of DNA is not unique because the sisters are still together, and the sisters are mostly identical to each other. They're not completely identical because of crossing over, but they're mostly identical. So here you would say that each of these are haploid. And another way of saying haploid is that you would say that the cells here are 1N. Now I put the 2 here in front because technically they have double 1N chromatids, or 46 chromatids, but they don't have 2N information. Okay, so this is sort of, again, like having 2 books that are the same book. That doesn't make 2 books of information. It's still 1 book of information. So you would call the cells 1N, but they are 2 times 1N pieces. Okay, it's a little bit confusing, but they are haploid at this point, which is why meiosis 1 is called reduction division, because we've reduced the amount of genetic information in each of these cells. So they are haploid, or 1N here. It's also important to note that these 2 cells, the one on the left and the one on the right, are non-identical to each other because of crossing over, and because one homolog went one way and the other homolog went the other way, and homologs are not identical to each other. So these 2 cells are not identical. If we replaced mitosis with this, we would mess the system up pretty darn quick, because we would be making daughter cells that are not the same as the parent cell, which you need in mitosis, right? You want to make always exact copies of things. And these are not exact copies. So that first split is called reduction division because we have 2 haploid cells. Each one has 2 haploid sets, but they're not a diploid set because they're mostly the same. So meiosis 1 is about separating homologs. That's what happens in meiosis 1. You take one homolog and send it in the left cell, and you take the other homolog and you send it in the right cell. And at this point, you'll probably be noticing that I'm using the word homolog and homologous pair a lot, and sister, and identical sister, and non-identical sister. I'm using that terminology a lot, which is why I try to set it straight at the beginning, because at this point, if you don't know what that means, it's going to be extremely confusing. Any questions at this point before we start digging into the second half? Mitosis, or meiosis 2 rather. Meiosis 2 is much simpler than this, thankfully, because a lot of the stuff that happens in meiosis 1 doesn't happen again. As Owen mentioned, it looks a lot like mitosis, which makes it way, way simpler. Question? Okay. Keep going here. And looking at meiosis 2. Oh no. Oh no. Oh, what is with... Oh boy, it's funny. It's funny that when you know I've used this note many times. It's funny what you notice all of a sudden. We are going to separate sisters here, but we're not going from 2n to n. We've already done that. The cells are 1n. We're just going to separate the two versions of n from each other. It's not 2n. It's 2 times 1n to 1 times 1n. Anyway, I just crossed that out. That doesn't make any sense. They're not 2n at the beginning of this. They are 1n cells. They have 1n genetic information. I really have to go back to my original note here and make some modifications, because that's... I can't believe I'm noticing this here for the first time. Anyway, we're separating sisters. That part is right. We are doing that. So interphase 2, and again, note the 2's in each stage here, looks very similar to mitosis, but there is one key difference. And that is you do not make a copy of the DNA. You don't copy the DNA again. When students show me this process, meiosis, for their project, so many students copy the DNA again in interphase 2. So many. And I always mention this, and they still do it anyway. Do not make another copy of the DNA. That part does not happen again. If you did that again, if you make another copy of the DNA, we will go back to having 92 pieces of DNA again. And then by the end, we're going to have cells with 46 pieces instead of 23 pieces. We're going to have too much DNA at the end. And then again, if you use that and put it together with another gamete, we're going to end up with a tetraploid zygote, which is fatal. You can't make a tetraploid zygote. It's not viable. So do not make another copy of the DNA, or busts the whole process. You do make a copy of centrioles and all of the stuff, all of the tools for doing cell division. So you do have to do all that stuff still, but do not copy the DNA. There basically is no S phase in interphase 2. Remember, S phase is synthesis. That's when you copy the DNA. So it skips that part. Okay, so we've made new centrioles, but other than that, we've done nothing. Okay, prophase 2 looks a lot like mitosis. Remember prophase? You take the DNA and you put it into chromosomes. So you're going to condense the DNA into chromatids. Oh boy, I better be careful with my vocabulary here. You're going to condense it into chromatids from chromatin. And you're going to bust up the nucleus. Get rid of that nucleus. That's what happens in prophase. But other than that, that's it. Is there tetrads forming? Nope, because there's no homologs, so you can't do that. Is there any kind of crossing over happening? Nope, none of that is happening in this process. So it looks a lot like mitosis. It's just DNA going from chromatin to chromatid, and the nucleus breaking down, and spindle fibers are forming from the centrioles, just like in mitosis. So it's way, way simpler. In metaphase 2, we're going to get the sisters lining up on the metaphase plate. Okay, this time they're going to go right on the plate, where each sister is on either side of the plate. Again, just like mitosis. This looks just like mitosis. Sisters are going to go right on the plate. Then, in anaphase 2, just like mitosis, we're going to pull the sisters apart. And one sister is going to go to the left, and one sister is going to go to the right, or top and bottom in this diagram, but same thing, one cell and the other cell. So we're going to yank them apart and put them in on opposite ends, just like mitosis. And then, at the very end, in telophase 2, and cytokinesis too, I kind of combined these together. You're going to put them into a new nucleus, and the DNA is going to decondense back into chromatin form again. But now, each individual cell at the end is going to have not only 1N information, but also 1N chromatids. That is 23 chromatids. So that is half of the number of chromatids that it would have had at the very beginning of this process, making a complete haploid cell. So that way you can take one of these that you make at the end, and then take another one from another person, mash them together, and you're going to get 46 chromatids, which would be the full genetic complement. Okay, so we have completed 1N cells here at the end. So what is really happening here, the purpose of this, is that we're going to separate sisters. Sisters get separated in meiosis too. Meiosis 1 is about separating homologs, and meiosis 1 is about separating homologs, and meiosis 2 is about separating sisters. So going way back, am I going too fast? Sorry if I am. This part looks exactly like mitosis, so it's easier to skip across it pretty quick. So at this point, you might be thinking, how come in females you only get one cell? Doesn't it make 4? I mean, you can see how it works in males. You get 4 cells at the end, 4 sperm. But in females, you only get 1 ootid at the end, 1 egg, instead of getting 4 eggs. You're like, well, what the heck? Why do you only get 1 egg? Good question. Good question, I ask myself. Absolutely there is, Hannah. If on Brightspace, if you click the button across the top that says course notes, or it's either called completed course notes or course notes, there is a completed PDF of every course note in there. So yes, I can double check that, but I'm fairly confident it's there. 3U course notes. Yep, they're all there. And I'll update what's in that folder as well, because I made some small changes to this one. I crossed some stuff out and whatever, so I'll add that in. As soon as we're done here, I'll upload it and make sure it's the same copy. But that's there right now. Do you know what I mean? It's on if you go to the top here, Brightspace, it's that thing that says 3U course notes. Oh, okay. All right. Cool. All right. So how is this different for females than it is for males? Well, as I mentioned, for males, these 4 cells will become 4 sperm cells. So that's pretty straightforward. You do this exact thing, and each one of those cells at the end becomes a sperm. In females, you still make 4 cells at the end. However, one of those cells is going to become the egg, and the other 3 cells are going to be called become what are called polar bodies. Okay, so there's 1 egg and 3 polar bodies. What a polar body is basically a nothing, a cell that doesn't have the necessary stuff to become an egg. And what happens to those cells is they do apoptosis. Apoptosis is cell suicide, basically. They break themselves down, and they give their nutrients to the surrounding cells in an organized way. Okay, they're basically useless cells, so they just pack up shop, shut her down. Why does that happen? We're actually going to get into this in a little bit, but the reason why this happens is that an egg cell, an ootid for humans, is gargantuan. It is the largest cell that the human body ever produces. It's so large that an ootid can be seen with the naked eye. That's absolutely ridiculous. It's around 0.3 of a millimeter in diameter. I think I have that right. 0.3? Or is it point? It's not 0.3, or is it 0.03 millimeters? Either way, this cell is big. No, it can't be 3/10 of a millimeter. That would be absolutely huge. Maybe it's 0.03 millimeters. So it's 3/300ths of a millimeter. But that's still visible to the naked eye. It looks like a little dot on a piece of paper. You can physically see an ootid. It's that big. Because the female part, the egg part, has to contain all of the machinery that's necessary to do, I don't know, thousands and thousands of cell divisions, all ready to go. Because remember that it's not receiving any resources from the mother for a while after it's fertilized. After it's fertilized, it has to do a whole bunch of cell divisions first before it can attach to the inside of the womb and start accepting nutrients and allowing the blastocyst to take nutrients from the mother. So it has to be ready to go to do thousands of cell divisions right from the get-go. So it's got to be basically gigantic. So in order to do that, there are uneven cell divisions. I mentioned billions of possibilities. We're going to talk about the number of actual possibilities genetically in a minute, but imagine you start off with an oocyte, and every time you do a cytokinesis, which is what I'm showing underneath here, so every time you do a cytokinesis or a split, it's an uneven split. So almost everything in the cell is going to go one way in the split, and just a teeny tiny little bit of stuff plus the DNA is going to go the other way. So you get a micro cell going one way and a gigantic cell going the other way. And then in the second split, which still happens, it's basically like on one side it's just a microcell splitting into two microcells, and on the other side you get another uneven split over here where virtually everything goes one way in the split and only a teeny tiny little bit goes the other way in the split. So at the end here you end up with 3 teeny tiny cells, which are called polar bodies. They're non-functional and cannot become a fertilized egg. And then you get one ootid, which is a gigantic cell that is going to become the egg cell in the end. The egg cell is like 100 times as large as the sperm. So we'll talk about female and male strategies later, but essentially females have to make gigantic cells in order for this process to work. Visible to the naked eye. I can't overemphasize how crazy that is, that this is a cell that is so big that you can look at it and be like, huh, there's a cell. That's crazy. That's so big. In sperm, the process is completely balanced. So you start off with a spermatocyte, which is a lot smaller than an oocyte to begin with. But then every time you split it, it's an even split. So you end up with 4 evenly split spermatozoa at the end. They're all the same size. And that's the main difference between this, which is that each split is going to be uneven in females, and in males, the splits are relatively even. And that's it. You get 4 at the end for males and one functional one at the end for females. Okay, that is the process of meiosis. We're going to talk about the implications of this in a second. We're going to talk about this idea of, well, I can mention this right here. I'll mention this. And then we're going to talk about sort of male and female strategy in a bit, and the difference between these cells and humans when we come back. And then we're going to spend some time talking about what happens when this process does not work properly, when it's broken. Because errors can occur during this process and all kinds of wacky stuff can happen. So we're going to talk about what happens when this process breaks when we come back after the break at 11:05. But just to quickly mention here, the 3 places where we create diversity, and remember diversity equals survival, so this is important, is we create it during crossing over, switching up DNA between homologs. We do it during independent assortment, which is where you decide maternal and paternal, or maternal and paternal, switch, or switch, all the way down the metaphase plate. Which way they go is totally random. That's independent assortment, so that increases diversity. And the last place where you get a randomness occurring is gamete pairing. So that basically what that means is which sperm is going to make it to the egg. And which sperm makes it to the egg is random. It's not a competition of the best sperm making it. the best sperm doesn't always make it. Well, certainly the best sperm doesn't make it because it's completely random. So it's millions of sperm are on the move, and which one actually makes it to the egg is just random. And because it's a random pairing, that increases diversity, because it's going to be, because even if you did that exact process 10 times in a row with the same sperm, and they're never the same sperm, but assume that they were the same sperm, it's not going to be the same sperm that makes it to the egg every time. It's random. So anytime you add randomness, you're going to increase diversity in the process. So these three things increase genetic diversity in sexually reproducing organisms, and again, lead to better survival for the organism. So this is a survival mechanism. We'll talk about that more in the evolution unit, why this is so important, but we've touched on it in diversity. Okay, I'm going to stop talking. I'm over time actually by a minute, and you guys go and stretch your legs, digest this craziness that we've just discussed together, and we're going to talk about some problems with it when I come back. All right, enjoy your break. So what we're going to do now is talk a little bit about the actual numbers in terms of how much diversity can be created from this process. Sorry, this is just something I added in here. We're probably going to do a little discussion together. I'm going to skip over this part because I mentioned all of this stuff during our little run through of meiosis. This is just the idea that during synapsis, you can get this swap occurring between homologs, and it just means that all of the sisters are going to have different combinations of alleles at the end. I've already mentioned this idea that basically no two chromatids will be the same at the end. Okay, that's all that this is saying. So it's mentioning here that that's the impact of crossing over. It means that every chromatid will be separate at the end. And then here's the idea here of, okay, if you don't have independent assortment, basically you get all paternal in some alleles, or in some gametes, and you get all maternal in some gametes. But with independent assortment, you get a variable combination of maternal and paternal in every gamete, and it just means that all of the combinations are going to be different of chromatids. So independent assortment leads to that. We already kind of went over the idea of that. So both of those things are contributing to this idea of genetic variation. And so it's actually quite difficult to determine how much diversity actually comes from crossing over, because there are so many potential domains or loci. Those are places on the chromosomes that can actually do the crossing over event. And you can either do it or not do it at all of those different loci. And whether or not you do the actual crossing over switch is random. So if you can imagine, if there are 200 locations on chromosome 1 where a crossing over event can happen, it either is or is not going to happen at each of those locations, and whether or not it happens is random. And then that's just for one chromosome. And then you look at chromosome 2, chromosome 3, all the way up to 22. It does not happen, by the way, on chromosome 23. So chromosome 23 is the sex chromosomes, if you'll remember, and they're not always identical, the sex chromosomes, because if it's an X and a Y, they're not going to be homologs to each other. X and Y chromosomes actually look completely different. They're not homologous. So you don't get crossing over happening on sex chromosomes. So it only happens up to chromosome 22 for humans. And for other species, it would not include the sex chromosomes. You don't get crossing over on sex chromosomes, but it happens on all the other ones. So, and that has some interesting repercussions for diversity with regards to sexual characteristics, but we're not really going to look at that in this course. But anyway, lots of potential diversity comes from crossing over. Because we have independent assortment is a little bit easier to calculate, because there are 23 pairs of homologs, and each pair could go this way or this way, where maternal goes left and paternal goes right, or opposite. So there's 2 options for 23 different pairs. And if they're random, that means that there are 8 million possible assortments during metaphase 1. Now keep in mind that metaphase is where you actually line them up on the left or the right. Anaphase is where they actually separate, but we say that the independent assortment occurs during metaphase because that's where they actually get positioned on the left or the right. So technically the sorting occurs before they get pulled apart, but and so that's why I'm saying metaphase. So in metaphase 1, there are 8 million possible combinations in humans that come just from independent assortment. And that's quite a bit. And if you look at gamete pairing, if the egg has 8 million possible combinations, and the sperm has 8 million possible combinations, and this is just from independent assortment, right? Then together, through gamete pairing, because it's random which ones encounter each other, that alone creates 64 trillion possible combinations of alleles from mother and father. And that is just for independent assortment and gamete pairing. So just those 2 things result in 64 trillion possible combinations. That's not even including all of the diversity that comes from crossing over, which is astronomical how many different possible combinations can be created in the crossing over process. So, I mean, the bare minimum, even if you didn't even have crossing over, is that you would get 64 trillion possible combinations. But then once you factor in the crossing over potential, you'd multiply them together. That times 64 trillion possible combinations. I mean, even if it's a small number, it would be astronomical, right? But it's not a small number. It's a huge number. So the end product of this is essentially you will never create the same baby twice, as long as they're not identical twins, and that's a different process that's happening there. It's one egg being fertilized and then separating into 2 zygotes later in the process, or 2 blastocysts, I guess, after there's been several divisions. So, yeah, there is a lot of potential variety here. You're not going to make the same baby twice is essentially the thing. So one last thing that I want to mention about this process is the different strategies that males and females undertake with regards to how they approach the process of sexual reproduction. So I'm going to talk about the differences here between sperm and egg, and that's really descriptive of what the strategies are for male and female, keeping in mind that these strategies are generally maintained in every animal. They're not necessarily maintained in every kingdom, but in general, they are. So again, there's exceptions to everything, but in general, when you look at the 2 strategies between males and females, the female strategy is quality. Build a quality product, okay? And that usually, if you focus on quality, that means you don't make a lot of quantity. So it's relatively low quantity and very high quality. You need to provide a lot of machinery and do it right, but you don't do a lot of it, okay? Whereas the male reproductive strategy is quantity. It's make a lot, not necessarily the highest quality, okay? So you'll notice this when we look at the stats here in terms of what males and females are doing. And again, this is extended to most of the animal kingdom as well, is that females are focusing on quality and males are focusing on quantity. So when we look at the number, first of all, of eggs produced, there's approximately 1 per menstrual cycle. I added in the approximate sign because it's not true to say that there's only 1 per menstrual cycle. I mean, in humans, there's usually 1, but it's very frequently 2. So, and it's very possible to have 2. That's how you get fraternal twins, by the way, that are not identical, is 2 eggs or more being released during a menstrual cycle. But it happens all the time. So it's really not just exclusively 1. It's basically like an average of 1, but there's usually more than 1. So about 1 per menstrual cycle, whereas males per ejaculation are producing 250 to 280 million, and that's if you have a fertile male. Sure, plenty of males are producing significantly less than that if they're having fertility problems, but that's a huge number. Okay, so we're talking about like 1 per approximately month versus 300 million-ish per ejaculation. Okay, so we're talking about a lot of potential sperm here is being produced. That's a quantity strategy based on quantity. When you're building an egg, oh, so I was actually a little bit high on my estimate for egg size. I forgot what the exact number was. 0.12 millimeters. So I said 0.3 millimeters, or 3/100ths of a millimeter, but it's actually closer to 1/100th of a millimeter. That's still visible to the naked eye, but it is quite small. I mean, it's not going to look like a giant egg. It's not like a chicken egg, but that is visible to the human eye still. So it's about 0.12 millimeters in diameter, whereas sperm are about 50 micrometers in diameter. I mean, we're talking about hundreds of times smaller than an egg. Thousands of times smaller, 10,000 times smaller than an egg. So obviously there's a trade-off here. If you want to make millions, they got to be smaller. Otherwise, the resources aren't going to be there in order to do that. And so if you're going to make one, then you have the option to make it bigger, which the females are producing all of the machinery necessary for the first many cell divisions, and which is why it needs to be bigger. In terms of ploidy, we already know that they're haploid, right? Sperm and egg are both going to be haploid. But females' eggs are locked in meiosis 2. I believe it's metaphase of meiosis 2. So you are born, females, with your full set of eggs. You have them all already. You're not doing the beginning parts of meiosis as an adult human. You've already done that when you're born. The cells that exist, the oocytes in your ovaries, already exist when you're born. They've already gone through meiosis 1. So it's the second part, meiosis 2, that gets completed during an ovulation or through a menstrual cycle. So there is a consequence to that. And that is that your eggs are already existing inside of you for your entire life. And one of the complications of that, the consequences of that, is that they are exposed to everything you are exposed to for your whole life. So radiation, which is just, there's just background, X-ray. They're exposed to every chemical that you ingest for your entire life. So as you age, your eggs become less and less viable over time, and they become more prone to errors, which we're going to talk about in a minute here. But that's a consequence of the fact that you've already produced all of your eggs. They're all already there. Whereas for males, it's also haploid. Obviously, they're producing haploid gametes, but they are producing them continuously for their entire life. So it's new cell lines are being produced, both by mitosis, because you got to make more spermatocytes, and also through meiosis, where you go with the spermatocytes and create spermatozoa. So those are new cell lines that are being produced over the course of your lifetime if you're a male. And so they're not subject to the same issues of deterioration over time as female cells are, which is probably one of the reasons why female fertility has a time limit. There is a time where the body allows you to be fertile, and then there's a time where you're no longer fertile during menopause or after menopause, whereas males are fertile for their entire life. So that's one advantage to producing all the cells as you go. But the disadvantage for males is that they don't get to contribute the same machinery to the process. I guess, I don't know if you call that disadvantage, but mitochondria and stuff like that, those all come from pretty much from the female side. In an egg cell, there are a lot of mitochondria, because keep in mind, you got to power all those cell divisions at the beginning, and you don't have the time or the materials to build a ton of mitochondria during that process. You got to power all these cellular divisions and stuff. So there's about 200,000-ish mitochondria inside an egg cell. Whereas in a sperm cell, there's around 100. Okay, so a lot less. And those mitochondria are only used for propelling the sperm. They produce energy for the flagella to operate, and that is all. So they really don't need very many. Now, that being said, those mitochondria that are inside the sperm do not actually get incorporated to the zygote, to the fertilized egg. They are not used in the fertilized egg. Only the DNA portion from the sperm is used. So when you look at your mitochondria that you have in all of your cells, they are made from your mother's mitochondria. You have none of your father's mitochondria in your cells. And mitochondria, really interestingly, have their own DNA. They have mitochondrial DNA. They do their own cell dividing inside. It's like a mini version of mitosis, the mitochondria do. And so you can actually use this you can decode the mitochondrial DNA in somebody, their mDNA. And through that process, you can trace back maternal family lines because you only have your mother's mitochondrial DNA, and your mother only has her mother's mitochondrial DNA, and her mother only has her mother's mitochondrial DNA. So and that DNA is virtually unchanged as you go up through maternal lines. So you can use that to figure out a lot of information about family trees. Because the mitochondrial DNA is unchanged through the maternal line. Kind of cool. I mean, it does get altered due to random genetic mutation and stuff like that, but it's largely the same. There are some special structural features here. There is an outer glycoprotein coat called the zona pellucida around the egg. And that glycoprotein coat only allows one single sperm to enter. And then it changes the shape of the glycoprotein coat and no longer allows sperm to enter, which is really cool. And in order for that to function properly, once a sperm comes in contact with that coating, it needs to change conformation around the entirety of the egg cell virtually instantaneously. Because if it accidentally allows two sperm to enter, that is essentially fatal because at that point there's going to be a triploid organism that's forming, which will not be viable. So it's pretty cool how that works. For males, people are already aware of this. Sperm have a tail for propulsion. It's not really as functional as you might think it is. A lot of the actual sperm movement that occurs in females is more of a result of the female moving and a lot less from the sperm swimming. So the mobility is limited. They're not swimming the entire distance, let's say. And they have some mitochondria in the middle for energy. They also have a little packet of enzymes called the acrosome at the front, and that is what interacts with the outer glycoprotein coat, which allows the sperm to enter into the egg cell. And there's some pictures of them at the bottom right there. And so you can see that the strategy between these two is quite different. You've got a quality cell with all the machinery. In the female, it's large. You make very few of them. And then the male idea is, let's strap some DNA to a missile and fire it and just hope it hits the target. That's a completely different strategy. Here you're focused on building a quality organism at the end, and here you're focused on simply getting the genetic material to the correct location. It's two different strategies. And so that general concept of that male and female strategy is replicated among almost everything in the animal kingdom and outside of the kingdoms as well. So certainly not in bacteria that don't sexually reproduce, but in other sexually reproducing protists, plants, there are similar strategies to these. Okay. Don't worry about these problems with fertilization. We don't have time for that. I did that previously. We talked about in vitro and stuff like that in the course previously. We don't have time, so we're going to skip over that. what we're going to do now is we're going to talk about what happens when this goes wrong. So, and the ways in which it can go wrong. So you'll need the next note package, which I believe is on the subsequent page. What are we doing here? It's on the sidebar here. Unit 2, Part 3, Genetic Processes course notes. There it is. Probably should put a link to it right in there, but I didn't. Towards the end of today, I was hoping that we would get a chance to start talking about inheritance, but you know what? I don't think that we're going to get to Mendelian inheritance, and we're going to have to do it at the beginning of tomorrow. By tomorrow, I mean Monday. Yay! It's the weekend. That's fun. All right, hopefully everybody's got those notes open at this point. Let's look at some problems. Okay, so I'm actually going to skip past this part here. Normally I would do this review of karyotypes because the last time we would have talked about karyotypes would have been 2 weeks ago in the normal course. Maybe I'll very, very briefly talk about this, but we just talked about this. This idea that when you take a sample from the amniotic fluid, you can lock the cell, a cell, in metaphase. You can blow it up, take a picture of the DNA with a microscope, take a picture of the chromosomes with a microscope, and you can organize them by size, banding pattern, centromere location. And they're organized from longest to shortest, and then you put the sex chromosomes at the end. That's a karyotype. Okay, so a karyotype is basically just a picture of the chromosomes. And the reason why I bring this up again is because this is how we used to diagnose a lot of these genetic problems that we're about to talk about. We didn't have sequencing, so there was no way to run the sequence of these and say, okay, well, you have the gene for cystic fibrosis. You have the gene for breast cancer. We didn't have that. That technology didn't exist until relatively recently, the last 15, 20 years. Which, I mean, I guess that seems like a long time ago to you. It's before you were born. But I mean, it's within my lifetime. So I see that as recent. So previous to that, we didn't even have the technology to look at anything specific. But we did do this. So we were able to look at the chromosomes and just check, are they all there? I mean, that's a good tool, which is to look at them and say, okay, do we have 2 of 1? Do we have 2 of 2? 2 of 3? All the way through to the end. Do we have 2 X's or an X and a Y? Or are we missing 1? Or do we have 3 of 1? All problems. Okay? So that was the strategy that we used to use. A lot of this karyotyping information can be done through a maternal blood test now. I don't think it's covered by OHIP, so you have to pay extra for this. It might be like $1,000 or something like that. You can have karyotyping done. That is covered by OHIP, I believe, but there are some risks associated with it, which is that you have to puncture the amniotic sac in order to remove a little bit of amniotic fluid. I mean, it's relatively low risk. We do it all the time, but when there's a risk associated with it and it's babies, people are extremely risk averse, and so generally people opt for the genetic testing, which you can get done from a maternal blood sample. So that's how it's different now than it was maybe 10 years ago. 10 years ago, there was no maternal blood sampling test that existed. So if you wanted to check, you had to do this. But anyway, what can happen is that during independent assortment and anaphase, remember anaphase is where you're going to take the two pairs of sisters, or the homologous pair, and you're going to pull them apart, right? That's during anaphase 1. You're going to yank these. They're in a tetrad, and you pull them apart, right? Or if it's happening during anaphase 2, you've got your sisters, and you're going to pull your sisters apart. That's what you want to happen during anaphase. I'm assuming you guys recall this. So we were just talking about this. So what can happen during this process is that when you try and pull them apart, the homologs stick together. They don't release each other the way they're supposed to release each other. Okay, and you end up getting this occurring. Okay, in other words, both going in the same direction. Or during anaphase 2, that would look like this. All right, so if both things are going in one direction during an anaphase, that's what's known as a non-disjunction. And I made a little correction in the notes. I used to write non-disjunction as 2 words. I was sure that it was 2 words. It's actually 1 word. So there's no hyphen in the middle. Non-disjunction is 1 word. That splitting, where one goes one way and the other one goes the other way, is called disjunction. It's called disjunction because you are breaking a junction between the two. So when disjunction does not happen, it's called non-disjunction. So non-disjunction is when you don't get a disjunction, when they don't break up apart from each other and both of them go in a single direction. So we're going to look at the 2 ways where non-disjunction can happen and what the outcome is in the gamete, and the outcome that happens eventually in the zygote, the fertilized zygote. Okay, so there's 2 ways this can happen, as I mentioned right here, which is that it can happen either during anaphase 1, and then it looks like this. That's what it looks like if it's anaphase 1. And if it's in anaphase 2, remember you're separating sisters, so it's going to look like this if it's in anaphase 2. Okay, so what is the end effect of that? So you've got your first division happening. This is an anaphase 1 non-disjunction. Okay, so these 2 would be homologs. I'm gonna color code them on my copy because it makes it easier to tell what's going on. So we've got paternal here, let's say, and maternal. All right, and they're in a tetrad, and then you try and yank them apart, but it doesn't work, and both of them end up here, going one direction in the split. Now you can assume that the other chromosomes had regular disjunction, and they split up normally. So this is only one of the homologous pairs where this didn't happen. If you get it with multiple ones, you're going to get apoptosis. And again, apoptosis is like programmed cell death. The cell is going to recognize that there has been a crazy thing that has occurred and a crazy subdivision of genetic information. It's going to cause apoptosis. So you generally don't get these really large multiple non-disjunction events occurring because they're fatal. So they result in a cell that is not viable. So sometimes it is viable though. So we're going to get this split where this is happening, and then we're going to end up with 2 gametes that are completely missing chromosome 1 or chromosome 2. They just don't have a chromosome 1 in them. Okay? Because remember that you go from 1 cell at the beginning to 4 cells at the end. So 2 of them are going to be blanks, and then the other 2 are going to be, well, they're diploid is what they are. They're diploid sperm, where they're diploid for 1 chromosome. Remember, you don't want them to be diploid here. You want the sperm or egg to be haploid. So we're going to end up with 2 here that are incorrect. Okay, so if you remember that the incorrect number of chromosomes, the word for that is aneuploidy. Okay, so this is an aneuploidy happening here, where there's the wrong number of chromosomes in these gametes. And it could be sperm or egg, so I'm keeping it wide open. So if I take now a normal gamete from another partner, okay, and I'm only looking at one of the chromosomes here, but let's say I bring in a normal gamete from another partner, all right? It could be a sperm or an egg, depending on whether this is male or female that's happening here. And I bring it in, and I merge it with this one, or I merge it with this one, or I merge it with this one. That's what's happening in the process, right? I'm going to merge them and make a zygote. Well, these are the 4 potential things that could happen. So there's my haploid gamete right there. Okay. So when I merge them, there are 4 different zygotes that could happen. So these are the possible zygotes. So you're either going to get one that's trisomic, okay, if it merges with the first one. That's trisomic means 3 of a particular chromosome. You don't want that. Or trisomic if it merges with the second one. Or, remember, it's completely missing one from one of the gametes for the last 2 here, for these 2. And so you're going to end up with only one of a particular chromosome in the zygote. That's monosomic or monosomic. Okay? So there's the options here is that you either get trisomic or monosomic if you have a non-disjunction that is happening in the first split. So the first division is in anaphase 1. Okay? So in anaphase 1, if they both go the same direction, you're going to end up with sperm that look like this, or eggs, potentially. Remember that 3 of the 4 wouldn't be viable anyway because it'd be polar bodies. But again, I'm not trying to confuse you here. Let's just talk about them as sperm then. 2 of the 4 sperm are going to have extra, and 2 of the 4 sperm are going to have nothing for a particular chromosome. Okay, and then when we get our zygotes at the end, once they get fertilized, these are the possible options that we're going to see. So that is a non-disjunction happening in the first anaphase. Non-disjunction, by the way, typically happens in the egg. And the reason that it's happening on the female end, it can happen for males too, but it's far less likely. Part of the reasoning behind that is believed to be because the eggs are sitting around. They are there for a long period of time with environmental exposure, and damage to the chromosomes is what causes non-disjunction events to happen. So there's oxidative damage or radiation damage or whatever to the potential oocytes, which is what's causing this non-disjunction event to occur. The other thing that could potentially happen, and I'll color code this one as well because we're just going to compare them here, is you could have, okay, we got our maternal and paternal here. So you've got a normal disjunction happening for the first anaphase. So this is good. So we had paternal go this way and maternal go this way. This is what's supposed to happen. But then in the second division, that's anaphase 2, we're going to get a non-disjunction event happening in one of them. Okay, usually it doesn't happen in both. That'd be a complete coincidence if it happened in both. But you got normal disjunction happening in the first one, and then the second one here has a non-disjunction where we've had, this one get pulled here, basically like this. Okay, so these are the gametes that you're going to potentially form in that situation. You're going to have 2 normal, okay, because normal disjunction happened on this side, but then on this side we had one that had an extra and one that had none. Okay, so again, if we take a sperm that is normal, okay, so would be, just draw it off to the side here. No, I won't draw it on the side. Right now my pencil is starting to get angry at me. Okay, so let's say we have a normal sperm here. And it's going to merge with these and form zygotes, right? So if it merges with this one, diploid, that's normal, right? This is a normal gamete. If it merges with this one, diploid, normal gamete. If the sperm merges with this one, we're going to have 3. So that's going to be triploid, bad. Or if it merges with this one, it's only going to have 1 of this chromosome, and that's going to be monoploid, also bad. Okay, so so here are the potential outcomes that you could get. So you could get so what we're looking at here are, again, potential zygotes. I got to restart my app here because it's starting to conk out on me. excuse me. I'm just going to restart this. This thing is starting to go real slow. Come on. There we go. All right. So, as I mentioned, if you have the wrong number in a gamete, in a zygote, we call that state aneuploidy. That's going to be a good word to add to your vocabulary list. So you either have an extra or an omission. The vast number of aneuploidies are non-viable. So that is to say, if you're missing a chromosome 2 or missing a chromosome 8 or whatever, almost all of those are immediately fatal for a zygote. You don't have the right amount of genetic information, and so you're not going to be able to produce a viable offspring with those aneuploidies. They just simply don't make babies. However, there are some aneuploidies that are viable, where you have a baby still, and they are various degrees of severity in terms of how that is going to change the outcome of your pregnancy. And we're going to talk about a couple of those. So, but it is important to note that most aneuploidies are fatal. Miscarriage, which is what happens often if an aneuploidy occurs, although it's not the only cause of miscarriage, there's many, is extremely common. People don't realize this, that spontaneous abortion, when you have a miscarriage, happens in 2/3 of all fertilizations. So like an absolutely huge proportion of fertilization events end in a spontaneous abortion, which a miscarriage. And I think it's actually really important for people to know that, because when people have miscarriages, they happen all the time. Everybody feels like they did something wrong, first of all, or that it happened, it only happens to them, that they're alone, and it's really rare for that to happen, and then they just got like some rare event that happened, which is like a tragedy. I mean, it's sad. It is sad, because when people detect that they are pregnant, that's a celebration. I mean, people are excited, usually. And you make something out of it, right? You kind of sort of like build in your mind this idea of like what it will be like to have a baby. Everybody does that pretty much. And so it is difficult. It's an emotionally difficult process. But I think it's made worse by the fact that people don't understand how common it is. If you knew that 2/3 of all fertilizations were going to end in a spontaneous abortion, I think you probably would temper your expectations a little bit more. And you would maybe acknowledge when it happens that it is really just a normal part of life. It's so common. That doesn't take away all the pain of it, but it does maybe change the experience a little bit of it for people. So anyway, just something to consider, that you may be in that position at some point in your life, or you know of people that are in that position, and it's helpful to know that you're not alone, that is something that is extremely common. Anyway, let's talk about some disorders that are caused by this. Anyway, all that's summarized here is exactly what I said above, which is this idea that you could either get this or you could get this, depending on when you get the non-disjunction during Anaphase 1 or Anaphase 2. And I don't want to spend forever harping on this. So the ones that I'm going to talk about down here are known as the viable aneuploidies. And they're called that because babies are born with these. There are other ones where babies can be born, but they are usually result in a child that lives for a very short amount of time. And so some of them don't even have names, because they often lead to either spontaneous abortion or death so early in life that it's just not named, because it's unlikely. These things often don't make it to term. So, but these ones are generally survivable for some length of time. Now, Patau Syndrome is probably the most tragic of these, because typically the median survival for a baby with Patau is about 3 days, which, I mean, devastating. That's a really hard thing for a parent to deal with. you can see structural defects. There's poor neurological performance at birth for a baby. And when a Patau syndrome is trisomy 13. So trisomy means 3 of, tri, of chromosome 13. So if you have 3 13s instead of 2, that is what causes Patau syndrome. And it's pretty, like I said, it's a pretty devastating thing to have a baby born with Patau. But it does happen. And I've already mentioned this, but the likelihood of this happening to you increases the older you are as a female. And so that's why usually if you're giving birth over the age of 35, there is additional testing that is recommended to you normally for these specific things. Under the age of 35, it's fairly rare. We're talking about maybe 1 in 10,000. But the older that you get, the more likely it is for this to happen. And it actually starts to go a little bit exponential the higher you get. So when you start getting into your late 30s and your early 40s, then these become very problematic. They're much more common. And so the testing that's recommended to you is much more extensive because they become more likely to occur. Edwards syndrome is trisomy 18, when you have 3 of your 18th chromosome. This one also, unfortunately, has a low rate of survival, although it is pretty variable in terms of how long the baby survives, depending on how severe the abnormalities are. But often there are severe problems with heart formation, kidney malformation, which makes your renal system not function properly, and other internal organ malformations, liver. And because of that, obviously that can really significantly affect the lifespan of an individual. So this one doesn't have quite as low rate of survival or a low median survival as Patau, but it's still quite significant. So so when I say that these are the survivable aneuploidies, I mean they're not necessarily survivable for a long time, but they are survivable for some length of time. Now, the one that you're probably most familiar with is trisomy 21, which is known as Down syndrome, because this is the most survivable one. And so you will meet people in your life that have trisomy 21, that have Down syndrome. And the way that Down syndrome is expressed is incredibly variable. This is really interesting in that some people can have relatively minor effects from Down syndrome, relatively minor cognitive impairment, relatively minor differences in facial characteristics and growth. It's usually still identifiable, but it can be rather minor. And then there's also, on the other side of the scale, you can have very profound effects from trisomy 21, where you have very serious cognitive impairments, and you can see organ malformation, growth impairment, and things like that. So there's quite a range that can happen in trisomy 21. The median lifetime the median lifespan of somebody with trisomy 21 used to be so low. It used to be like 18 or something, but it's much, much higher now. And the reason for that is we're just able to treat a lot of the effects of trisomy 21 a lot better, like a lot of the organ problems and stuff like that associated with it. We have much better treatments for those things. So the median life expectancy is much, much longer. It's not the same as the general population, but it's considerably longer than it used to be. So it's not quite as significant in terms of risk of death. Obviously, the effects of it are significant. But anyway, it's definitely the most survivable of the autosomal aneuploidies. So autosomal, there's that term again. An autosome, remember, are the non-sex chromosomes. They're pretty important. So when you get an aneuploidy of your autosomes, most of them are fatal. Here's some examples of some ones that are potentially survivable for some length of time. But autosomes do not appreciate having too much or too little genetic information. Notice that these are all trisomies. I don't know of a single survivable monosomy. I don't think any of them are survivable. So monosomy is, as far as I know, always fatal. Although, correct me if I'm wrong, maybe you find an example of a monosomy that is survivable for humans. I don't know of any. Whereas sex chromosomes, because they are primarily responsible for secondary sexual characteristics, there are a number of aneuploidies that can exist in sex chromosomes or allosomes. Remember, these are sex chromosomes are called allosomes, so these would be allosomal aneuploidies. There are a lot more of these that are potentially survivable. Some of them are even hidden. Like if you met somebody that had this aneuploidy, you wouldn't even know. So because they have relatively low effects. So I mean, depending on the one. So let's look at a couple of them really quickly here. Turner syndrome is a monosomy, and I did mention that monosomies are not survivable in allosomes, or sorry, not in allosomes, in autosomes. In allosomes, they are survivable because again, it's not as critical here. So monosomy X means you have an X, but you don't have a second sex chromosome, X or Y. Now keep in mind that males also only have an X, and females don't have a Y. So one X is obviously survivable. All males have that. And not having a Y is also survivable because all females have that. So this is a hybrid of not having the thing that females have and not having the thing that males have. It is survivable. It does often come along with some characteristic physical abnormalities like short stature. So people are typically shorter. They have a broad chest, a lower hairline in the front, lower set ears than is typical. Some of them are born with webbed necks. They are biologically female in that remember that female's the default. I mentioned that way back here. So if you've got an X and no Y, you're going to be biologically female because you don't have the female's the default, and so you don't have any of the signaling pathways from the Y that are going to make you become a male. So you will be biologically female. But you usually have some type of gonadal dysfunction, which means that your ovaries are not functioning correctly due to the lack of additional production of female sex hormones and things like that. Okay, and you also, because of the lack of those hormones, are often amenorrheic, which means that you do not have a menstrual cycle as a female, and it usually leads to sterility. I don't know of any cases of this that are where people are not sterile, so it's generally people are sterile if they have Turner syndrome. Sometimes this is more obvious in terms of physical features, and sometimes it's less. So that one's fairly variable. Klinefelter syndrome is when you have both of your X's, as a female would, plus you have a Y. So remember that you have the X. Anytime you have the X, you're going to get sent down that developmental pathway that leads to male sex. Okay? So you're going to be born as a male, and males have an X. They always have an X. But here you have an extra X. Okay? So you can think about what that would potentially do. We're talking about additional production of female sex hormones. We're talking about, and I mean, that is really the primary thing that is going to happen here. What does that lead to? Well, it leads to, again, gonadal dysfunction, smaller testicles, reduced fertility. It doesn't always mean infertility, but it does tend to impede the functionality of sperm production. But there aren't a whole lot of other effects that come along with Klinefelter syndrome. So it's very possible that you have Klinefelter syndrome as a male. You may never know. Maybe you have genetic testing later in life and you find out, oh, hey, I got an extra X chromosome. But it's quite possible for this to remain hidden in the population if you've never been tested, and it's certainly possible that you have Klinefelters. So this kind of stuff can be hidden in an individual and you wouldn't necessarily know it. Triple X syndrome, otherwise known as trisomy X, is when you have 3 X's. So this is also a possibility. There's no Y, so you're going to become biologically female in this case. And there are no physical features that are associated with it or medical problems. So here's another one that could be potentially just hidden in the population. As a female, you could have triple X syndrome. You wouldn't know. It does have some cognitive symptoms that are associated with it. Not always, and again, there's always a range of experiences for these, but it often results in things like learning disabilities and delayed speech, delayed use of language skills early in life. But you have normal sexual development. You're able to produce regular female sex hormones. You don't have gonadal dysfunction, so you're able to have a regular menstrual cycle. And so again, triple X syndrome is one of those things that can just be hidden in the population. The last one here, XYY syndrome, which is sometimes known as Superman syndrome. It's when you have the X and the Y like a regular male, and then you have extra Y, bonus Y. And remember that Y is what is responsible for the development of secondary sexual characteristics for males, and you're just doubling it up. So you got extra. And so you end up with things like extra production of growth hormone, extra production of testosterone. And so when you get that, you end up with a male that is often taller than average. There's not necessarily any unusual physical features. So you're not going to be able to pick out somebody from a crowd that has XYY syndrome. You get normal sexual development, again, because you have all the things that you need, but just a little bit of extra of secondary sexual characteristics for males. But really, this isn't that obvious. So again, you could be a male right now sitting there and have XYY syndrome. You'd never know. So these are kind of fascinating. These aneuploidies, the three here are kind of hidden. Turner syndrome has a little bit more that's associated with it, and so it tends to be more obvious in the population. But the last three here can easily be hidden. So yeah, I find that fascinating. So some aneuploidies are not necessarily so detrimental to human health, whereas if you have an autosomal aneuploidy, this is going to affect your life in some way. All right. So, and then we already talked about how you see this. You do an amnio, you look at the amniotic fluid, you look at the karyotype, and you can see these. And you can also determine these now through a maternal blood test. So, or at least your likelihood of getting them through a maternal blood test. If you get that maternal blood test and it returns a product that says you are likely to have one of these, then you get the amnio done and to confirm it. So you still have to get the amnio done if it's demanded by the maternal blood test. So that is if you have a non-disjunction event. That is one way to have the DNA be somehow incorrect in the gametes that you're going to produce. But there's another way. And the other way happens during crossing over. Okay, so during crossing over, or some of these are potentially happening during the S phase of interphase 1, when you're making your copy of your DNA. So some of these can happen at that point as well. Most of these are actually occurring during the crossing over, though. So some problems that can happen during crossing over, and remember that if you are altering the actual sequence of the DNA, we call that a mutation. Okay, so mutation is when you're changing the sequence. We talked about the idea of like ultraviolet radiation mutating or causing a change in the sequence. Obviously, that can happen as well. But that doesn't usually happen in your sperm or egg because they're not exposed to sunlight. I mean, I guess you could, be hanging out outside of a nuclear reactor or something like that and being exposed to gamma or something like that, and then having some of these mutations potentially occur in your gametes. But typically, this stuff is occurring during crossing over. By the way, I would have this open as a reference when you're doing your quiz, because I ask questions about identifying these. But anyway, deletion. By the way, all the first three here are single chromosome mutations that happen during crossing over, which means that they're going to happen to a chromosome. So they're going to happen to chromosome 1, or they're going to happen to chromosome 2, or happen to chromosome 3, or whatever. That's a single chromosome mutation. So what you could have is that while you're doing your crossing over, you know how you're supposed to take a piece and then you're supposed to switch it with a piece on the homolog? Well, what happens when you're doing the switch and that piece that's supposed to come back to your piece just gets thrown away? That happens. So when it just gets thrown away, it doesn't end up getting incorporated into you. Your chromosome is going to get shorter. So it was this long, and then now you've taken that part out and it's gotten shorter. You basically cut a piece of it out. So that's called a deletion. Obviously, ranges in severity depending on what you've deleted. But if you've deleted important things, it can be devastating. It really depends on what's been deleted. So that's one potential mutation that can occur. You can get a deletion. What can happen is when you're doing the crossing over event, an enzyme may come in contact with that piece and actually make a copy of that piece. So you end up with a double of it, and then the double is exchanged. It's also possible for the original piece to stay where it is. Oops. And then instead of having an even exchange, both pieces come over to the same piece of DNA. That's also a duplication. So that leads to a doubling up of a particular sequence. Sometimes duplication errors have no effect or a relatively minor effect, and sometimes they're very significant. So again, it depends on what you're duplicating. An inversion is where you're going to take a sequence, like let's say that you have a sequence of genes here that goes sequence A, sequence B, sequence C, D, E, F. And then when you're exchanging it, you accidentally flip it. Remember that the DNA can only be read in one direction, and it is possible to flip these and then reinsert them upside down. If you do that, it makes all of the DNA that you've switched over to the other side meaningless, because you can't read a book backwards. It doesn't work like that. And the same thing is true for DNA. It can only read in one direction. So if you do what's called an inversion, if you flip over a piece, it's broken. It no longer, that will be a non-readable part of the DNA after that point. So those are all known as single chromosome mutations. They're all happening on one chromosome here. There are also chromosome mutations that happen between two chromosomes. Okay, so this is when you get a crossing over event happening, but not between homologs. So in other words, you get a tetrad that is forming between chromosome 1 and chromosome 6. Those are not supposed to attach in a tetrad, but it happens. So you accidentally attach them in a tetrad, and maybe you get a switch of a DNA sequence from chromosome 4, and it gets moved over to chromosome 20. Okay? So now your chromosome 20 has a piece of your chromosome 4 in it. They've changed size, basically, because they've had this uneven switch between chromosomes that are not supposed to exchange. They're not homologous, so they're not supposed to exchange information between them. So that right there is called an insertion. You've taken a piece of 5 and put it on 21, or taken a piece of 15 and put it on number 6. And again, the effects of these are variable depending on what you've exchanged, but they can be very serious. And then the last here is that what if you actually get a complete switch between 4 and 20? Normally this is very difficult to do because what you're switching is completely different. But it does happen. So you might get a translocation. Translocation means you're just relocating a piece of 4 onto 20 and maybe moving a piece of 20 onto 4. So instead of just having a piece of 4 go to 20, you're actually switching a piece between 4 and 20. Those two are actually kind of similar, but one is where it's only going in one direction from one to the other, and translocation is when you're actually exchanging a piece between two different chromosomes. It has a different name. Okay, so obviously all of those things can potentially cause problems because your gamete at the end is going to have something unusual in it. Usually these result in apoptosis. They result in a non-viable, again, depending on the severity, they usually result in a non-viable birth. However, some of these do lead to viable offspring. So it is possible for genetic disorders to be based on these, especially if you have something like a very small deletion or a duplication. Oftentimes, if you're duplicating something relatively unimportant, this doesn't necessarily impact viability that much. So, which is interesting. Okay, what are you going to do with this? I'm actually going to give you the rest of the time to work. So first of all, there are some questions to go along with meiosis in general. They're in your textbook. It's page 160, 4, 5, 6, 7, and 11. And these are questions that are just reviewing the concept of meiosis. Once you've done that, I strongly encourage you to try the practice quiz, which remember does not count for marks, but it is going to give you a little bit of a review of your understanding of meiosis. So try that. And then there is a little bit to go along with errors. I've added in a short reading from the text, 162 to 166, and it talks about some other ways in which those errors can impact meiosis as well. You can skim a little bit, but that's kind of an important little bit of extra knowledge there on how errors are going to impact the process. And then there are some questions that go along with it from the text. 166, 1, 2, 3, 8, 6, and 9. All of these textbook questions are going to go in your homework document for Unit 2, which I don't think you've created yet. I don't think you've done any of the textbook questions yet. So you're going to need to create a homework document, and there's a little exit quiz that you do at the end, again, just to check your knowledge. So I'm going to give you the rest of class to work on this in Learning Block 3. You've probably run into some things you might want to add to your vocabulary list today. I assume you have. And the stuff that I've set up here for Block 2, this discussion of Mendelian inheritance, we are actually going to start tomorrow. Don't even worry about watching this video. I'm going to leave that for tomorrow. And by tomorrow, I mean Monday. I keep forgetting it's Friday. Before we move on, we covered a whole heck of a lot of stuff today. This unit is jam-packed with goodies. Are there any questions about meiosis in general? Or the errors that are possible, non-disjunction, deletion, insertion, translocation, etc.? Or just genetic disorders in general? If you're interested in striking up a larger conversation about genetic disorders or whatever, genetic testing, or whatever you want to talk about, stop by in the Google Meet. And then we'll talk about whatever you're interested in. Okay, I'm not seeing anything pop up over there. I'm not sure if I gave you enough time. I hope I did. If I do, I'll 100% answer your question. But as you're going, if you have any questions about the homework, about anything that we've covered today, please let me know in the chat. If you want to talk about something in more detail, drop by the Google Meet. For tomorrow, don't forget that you will need 2 colors of paper and some tape and maybe a pencil or a pen. Or you could use 2 colors of pipe cleaner. You need 4 pipe cleaners, 2 of one color and 2 of another color. Or, I mean, pretty much anything that you can use to represent chromosomes. We are going to cut the chromosomes to represent crossing over, and we're going to reconnect them. So that's what the tape is for. But if you can do that, if you can manipulate something to represent chromosomes, you're good to go. And you'll need at least 2 different colors of those things. So consider that. We could bring that for tomorrow. We're going to start tomorrow with a little bit of a review of meiosis. It's a very complex process, and usually it demands at least 1 review, just to make sure we're all on the same page. Okay, if you guys need anything, let me know. I'll see you in the Google Meet if you're interested, and if not, I will see you on Monday. Enjoy your weekend. You have done it.